Hello, everybody. As Andrew has said, my name is George Georgiou. I'm a curator for the Gene Ontology Annotation project as part of the protein function content team. Today, I'm going to be giving you an introduction to QuickGo, our online browser for the Gene Ontology and Gene Ontology Annotations. So to start off, what is the Gene Ontology? Well, it's a vocabulary, a controlled vocabulary that allows us to capture biological knowledge in a written and computable form. And it's a set of concepts, and the relationships to each other are arranged in a set of a hierarchy. So as you see on the slide here, we have this tree of terms. These are all Go terms, and they're going from less specific concepts at the top to more specific contents. So we have at the very top biological process, which is the parent term for this entire branch of the ontology, which has multiple children, all connected with different color arrows, which represent the different relationships between the terms themselves. What aspects are covered by the Go? Well, it's technically three structured ontologies that make up the gene ontology, and they talk about first biological processes that a gene product may be involved in, the molecular function that they may have, and where these functions and processes take place. So that's referred to as a cellular component. So for example, we can take a protein kinase, which would be involved in protein phosphorylation, which is the biological process. It would have a molecular function of kinase activity, and we'll say it's a cytosolic one. So the cellular component Go term you would use then is cytosol. So how is it structured? I said before that it's connected. These terms are connected by a series of relationships, and these relationships are represented by the different color arrows. We have a little legend here, but I'll go through them individually. So the first is the relationships are is-a, part of, regulates, positively and negatively regulates, occurs in, capable of, and capable of, part of. Is-a, the first relationship, is where you have a direct connection between the parent term and the child term. So for example, we give the example of the urea cycle here, the urea cycle, where it is the urea cycle is a type of urea metabolic process and is a type of amide biosynthetic process. So these two processes are the parent terms to the urea cycle. So they are less specific terms going to a more specific term. Part of is the next relationship. This one, we use the example of photosynthesis, where the photosynthetic dark and light reactions are part of photosynthesis, because photosynthesis, as we know, can be broken down to the light and dark reactions. So you see it here in the ontology. This is represented by a blue arrow. There are three regulates relationships. There's the regulates, positively and negatively regulates. So yellow represents the regulates, green for the positively regulates, and red for the negatively regulates. And the example we give here of how these relationships are used. So for the regulates, we say the regulation of the immune response regulates the immune response. So in other words, gene product in question, we are not able to really say whether it positively or negatively regulates. So we say it just has regulation of the immune response. Positively regulates, in this case, we say the immune response is positively regulated, and subsequently with the negatively regulates, we have the immune response is negatively regulated. So a gene product can either have one function or both, depending on the situation. Occurs in is one that we use for the cellular components ontology. So we say, for example, intracellular transport occurs in the intracellular compartment. So we connect the different ontologies where we can take the process term and connect it to a cellular component term. Capable of, in this case, we say, for example, the SAGA complex is capable of the thiol-dependent ubiquitin specific protease activity. So this complex is capable of having this molecular function. And then there's capable of, part of. This one's a little bit more confusing to understand because it is consisting of two different relationships merged into one, but when you see how we have it here, it's a little bit more obvious. So the example we use is the synapsis initiation complex, where we say it is capable of the regulation of synaptonemal complex assembly and homologous recombination and is part of the synapsis. So it is part of the synapsis, and as we see, it is capable of two processes. So what is the aim of the gene ontology? Well, we use it to describe the normal functions and processes and locations of gene products, and it does not capture pathological data, any processes or experimental, as well as experimental conditions or temporal information. Go is not used for disease annotation. In this case, we are interested only in the normal biology, what you would find in a wild type cell per se, or an animal. And you've been hearing me use this term gene product a lot. So what we refer to as a gene product is any molecule that is synthesized by what a gene encodes. In this case, we use gene ontology to describe RNA, proteins, and protein complexes. And the identifiers and databases that we pull these proteins and RNAs and protein complexes from, for RNAs, RNA Central, for proteins, UniProt, and from protein complexes, Complex Portal. So we use Go to describe these. So now that you have a brief understanding of how the ontology is structured and what we use it to describe, we're going to actually talk about what we output as part of the Go annotation project. And that is the actual annotations themselves. So what makes up an annotation? It's a statement about a gene product. And we usually say that it has a particular molecular function, or it's involved in a particular process, or it's located in a certain cellular compartment as determined by a particular method. So in this case, that is the evidence code over here on the bottom, if we're looking at the example here, and as described by a particular reference, which is a reference. So we have our gene product that has a particular activity, or it's involved in a process, or it's located somewhere in a cell, which is usually referenced to by the Go ID and the Go term name, as supported in this case, we use a PMID, so a paper from PubMed, and this is the type of evidence that supports it. With evidences, we actually use the evidence and conclusion ontology. We can't just make a statement without providing any kind of information about where we got it from. So we can't assign a Go term without an evidence and a reference. So we use ECO for evidence and conclusion ontology. So we say that Go term for protein X, in this case, we use the example of alcohol dehydrogenase activity, has an experiment that showed through direct assay, that's what IDA stands for, inferred by direct assay, from this fake PMID ID over here. And pretty much our job as curators is to read the papers, break them down into the Go annotations, and make sure that we reference them accordingly and provide the adequate, the correct experimental evidence to show how we got that annotation. So what classes of evidence code are used? So I said we use ECO, but this is something that's recent development. We've been using in the past Go evidence codes. So such things as direct assay, mutant phenotype, physical interaction, genetic interaction, expression. There's high throughput variants of those. We have traceable author statements, non-traceable author statements. So statements that are in papers that don't have a reference. There's sequence similarity, genomic context. There's a whole list here that we can go through, but what we do here, what I provided with you here is the ECO IDs. So when we get to QuickGo later, you can actually go and look up what types of evidence these refer to. But to give you an idea, direct assay, for example, children of direct assay, because ECO is also an ontology, so it has parent terms and children terms. ECO 314, which is our direct assay evidence, for example, kinase assays, in vitro reconstitution, immunofluorescence, cell fractionation. These are the type of evidences that we can use in Go annotation to show where we got the annotation from, or how we made the annotation rather. Something else that you should be aware of, and we're going to talk about it in a little bit later when we go through QuickGo itself, is what is a Go term qualifier. These are special terms that specify, well, so as it says here, it specifies a relationship between the gene product and the Go term in the annotation. It's to help us get more specificity in the annotation. And the ones that we currently are using in Go are listed off over here. I'm not going to read them all because it will just take a while, but if you're curious about these, there's more information at the link that I've provided at the bottom of the slide. And if I forgot to mention earlier in the webinar that the slides are already available for download in the materials section. Now, these qualifiers, they can change the relationship between the Go term, and sometimes you can also change that qualifier by including the expression not before it. So for example, not enables, so it does not have that particular molecular function, to indicate that a gene product does not do what you might expect it to do. So for example, we have a kinase in one species that we know is active and it can phosphorylate lots of different proteins, but in the orthologous kinase in another species is inactive. So we would actually put down that that particular kinase in human, for example, enables kinase activity. That gene product enables kinase activity. But let's say in C. elegans, for some reason, that kinase is inactive. Maybe it's a pseudokinase there, or it just is missing some, a pseudokinase, we'll go with that. And then we would actually put the not enables kinase activity as our annotation. Last, there are ways to actually make a Go annotation more specific, and that's to use the annotation extension. So it allows us to combine Go terms with other Go terms, terms from external ontologies like Uberon and KEBI, and as well as database identifiers representing genes or their products. So if we know something is going to be, in this case, we use the example of PAP1, where it's a transcription factor that regulates transcription of several oxidative stress response genes. Normally, the simple Go annotation would be PAP1 regulation of transcription. But with annotation extension, we can actually list off the targets that it is responsible for regulating transcription of, as highlighted here in the blue. And we use another example here, TMM115. It's located in the nucleus. That is part of an epithelial cell, so that we would get that from the Uberon ontology. That is part of the cervix epithelium. So we can make annotations even more specific. But the idea is that the extension, even if an annotation was not to have an extension, is just as true with the extension. So how can scientists use the gene ontology? Well, we can use it to access gene product functional information, analyze high throughput genomic or proteomic data sets, validate experimental techniques, such as cell fractionation. So if you want to use cellular component ontology for that in particular, get a broad overview of a proteome, and then also obtain functional information for novel gene products. Now, I'm not going to go through each and every one of these types of examples, but there was a previous webinar that we did where we did cover this, and I do suggest if you are curious about how to use Go for that particularly, to go check that out. So what is QuickGo? As I mentioned earlier, it's our online browser for Go, Go annotations, and also the eco. You can search for Go and eco terms as well as gene products. You can filter the annotation sets for annotations that you're particularly interested in, and you can use it to create or use preset Go slims. And I'm going to talk about Go slims at the end, so don't worry, I haven't mentioned it yet, but we will cover it. And we keep QuickGo as up to date as possible, so we update the annotations once a week, and we update the ontology daily. So this is our QuickGo homepage, so you can get to it at the link below, which is just ebi.ac.uk/QuickGo. So we're just going to work our way through it. So let's start with the search bar. So big red circle over here, that's our search bar. You can type in a general search term. So by keyword search, you can type in particular Go ID if you know the Go term ID you're looking for, the eco code if you know it in particular, or the name of a gene product. So for example, if we type in apoptosis, we see that a list appears. So we get terms and gene products. And if you were to actually click on the show all results for, let's say, terms, in this case, I search for kinase activity. What we get is a search page that looks like this. And it allows us to filter for the ontology aspects as well as break down the ontology types. So since we have two in QuickGo, which is Go and eco, you're able to distinguish them. And then we provide a link to the Go term definition page, an option to add the term to the basket or view the Go term in the ancestry tree. And then also, if you click on the annotations button to the right-hand side of it, it brings you to the annotation table where you can see all the annotations to that particular Go term and any of its children. So if we were to, let's say, go ahead and look at a Go term page, in this case, we're going to look at ATP binding. Let's actually take a look at what it's made up of. We have on the left-hand side our navigation bar that you can just click to get to the section that you're most interested in. We have an option to add the Go term to your basket, and also an option again to view all annotations to this Go term. And right below it, of course, and sorry about that, I didn't highlight it, is the ancestry tree for how that Go term is related to molecular function. So this is interactive. You can click on it and it will actually take you to the definition page for the Go term to one of its parent Go terms. We also have in the next, a little bit further down when you scroll down, the child terms. So all the child terms related to this one in particular. So we see over here, in this case, it is the ATP binding. So it could be capable of the mute S complex. So this child terms lists off the Go term as well as the relationship to the parent. Annotation guidelines, so guidelines on how this term should be used. So this is if you're curious about how an annotation, how a Go term is specifically used across multiple annotations, this is where you can go and learn a bit more about it using the link that's highlighted over there in blue, as well as the annotation blacklist. This is to show the restrictions that we've placed on this Go term for assigning it to orthologous species from electronic annotation. This is something that I'm not going to really go diving into because that is a whole nother webinar if you really want to learn about it. In addition to those, we have the cross-references and cross-ontology relations. So for example, this particular binding term has interpro family cross-references, as well as cross-ontology relations with the KEBI term over here for the term ATP. So that's the KEBI ID for ATP, so ATP binding would naturally have that. And then we also have co-occurring terms. So if you want to know what other terms are typically used for a gene product that has a function of ATP binding, this is a table where you would look. And finally, if you're really interested in the change log, which shows you the history of the Go term and any changes that have been made to it over time. I'm not sure really how often this would interest you, but if you are someone who's interested in looking and developing ontologies, this might be of use. So instead of clicking on the terms, let's say I clicked on the gene products, show all results. Now we see that we have a much bigger list for kinase activity again. So now we allow different filterings for the entries in UniProt, whether you want SwissProt or Tremble, SwissProt being the manually curated entries, Tremble being electronically generated ones. Filter for the type of gene product that might have this. So here we break it down on protein, complex, and microRNA. The organism, so if you know the gene product you're looking for is within human and you want annotations to, you want a kinase that's been annotated to kinase activity in, let's say, I don't know, zebrafish, you would use the filters for species. And then finally, proteome status. So whether you want something from the reference proteome or the complete proteome, which would include isoforms, that's where you would use that filter as well. And then of course, within the table itself, we provide a link to the database entry for the gene product. So you can click on that, it will actually take you to UniProt. And if you click on the annotation section, it will actually take you to the annotation table and show you all the annotations to that particular gene product. Going back to our home page, let's go on to the annotation viewer. So the annotation table, as you see from here, this is now, well, today we just had a release, so I didn't have a chance to update the screenshot, but as you see, we have over 556 million annotations. So it's going to be a little bit difficult to find exactly what you're looking for just scrolling through the pages. So in this case, what we've done is we've implemented filters to help you get through to find the annotations that you want. And that's what's highlighted in red there. So we'll go through each of them. The first one is taxon. So this is using taxonomy to find the species that you're interested in. We use taxon IDs, and if you're not sure of it, we have a link to the UniProt taxonomy page over here that you can use to search for your particular species. We've already listed off in the box here a lot of the model organisms. So if you're not looking at one of those, then you have to probably look up your taxon ID. We've also included an option to include descendants of that taxon ID. So if you're looking at something, let's say E. coli, but you don't know which strain that you're interested, in, you want to see all E. coli annotations, you would use the taxon ID for the general E. coli and then include descendants. And next after that is the gene products. So this is again allowing you to sort for annotations to either RNA, protein complexes, or proteins. Now, if you click on proteins, you get further options that appear, which show you the UniProt KB, so reviewed and unreviewed. So SwissProt being the reviewed, the manually curated entries, and unreviewed from the electronic, as well as selecting from whether you want annotations to the gene product specifically for the reference proteome or to the complete proteome, or the reference proteome with other isoforms as well. And of course, if you know your gene product ID, either using a RNA central identifier or a complex portal identifier or UniProt identifier, you can throw them in the little box over here and hit add and then apply, and you should be able to filter out for the gene products you're interested in. The next filter is the Go terms filter. So if you know the identifiers of your Go terms in particular, you would throw them in the box up here at the top. And then hit add. Or if you want to use them for a SLIM, which I'll go into later, you can throw in your term identifiers here or choose a predefined SLIM over here in this bar. But if you were to select it for a SLIM, you can then throw them in, select the SLIM option, hit apply. And if you click on the options in the Go terms filter, you actually get another little filter parameters that appear that allows you to kind of look at, and further specify the filter that you want to apply. So in this case, the default filters are include child terms of the Go term that you're interested in. So if you choose a very non-specific Go term like glycosylation, you'll actually get the children of it, like protein glycosylation, like glycosyltransferases and things like that. You have an option to use these terms as a Go SLIM, or use these terms as an exact match. So let's say you don't want annotations to the children, but just specifically to tyrosine kinase activity. So you would use these, you would select the third option. And then of course you can specify the relationship, which I went through earlier. These are is a part of occursin, and is a part of occursin and regulates. Our default option is a part of an occursin, the three basic qualifiers that we always include in the general annotation set. Next is the references. So if you happen to know the paper that you want, if you want to see if a paper's been curated, this is a good way to do it. So you throw in your PMID ID as shown in over here. If you're familiar with the Go consortium and how Go refs work, you can always throw in the Go ref to get annotations to that specific database for that particular Go ref. If you're interested in annotations from a particular reactome reaction, you can select the reactome option. There's several different identifiers you can use in the reference field. So most times though, our user base has been looking for annotations from papers. So these are ones that have been manually curated. So they've been looked at by a human and they've been added that way. So that's always the first option, is why we list it as any PubMed ID. Next is the aspect filter. This allows you to filter for the aspect in the ontology that you're interested in, of Go that you're interested in. So whether you're only interested in cellular component annotations or biological process annotations, this is where you would do that. Evidence, so those evidence code table I showed you before, we offer several different filters to go for that particular evidence code and any child of it, which is what the bottom over there would show you. Where it says include child terms or exact match. We also allow you to filter for all evidences used in manual assertion. So these are any that would be from manual curation and not from electronic curation, which electronic curation is represented by the IEA or ECO 256. That option is further down. So you can always choose what type of evidence you want to go for. Next is extensions. So you can filter for any annotation that has an extension. There's a little button hidden by this drop-down menu that says view all annotations with annotation extension. Or if you're particularly interested in, let's say, annotations that have an annotation extension to epithelial cells. You're only interested in gene products that have an annotation to epithelial cells. You can define the relationship over here in the first box. The database, so if you're interested in epithelial cells, it would be the Uberon database. There's another drop-down menu that appears when you click in there. And then you put in the identifier for epithelial cell from the Uberon ontology and then hit apply, and it will only show you those annotations. Finally, if you clicked on more, the last little filter, It shows you a table of the qualifier, the with from, and the assign by. The qualifier was those relationships that I mentioned earlier that modify that set the relationship between a gene product and its Go term. So you can actually specify that if you're interested in. And then, of course, the assign by is the database that has generated the Go terms. So if you're interested in only ones from Uniprot, you would scroll down from the ones to Uniprot and select those. Or if you're interested in ones from AgBase, you can select that right there. And the with from field is really more important for genetic interaction and for protein interaction for manual entries. Or if you see that an annotation's been made by sequence similarity, so an ISS evidence code, and you see it's to another database identifier, you can use the with from filter to specify the database identifier there. So next part, we're almost at the end, so don't worry. Hopefully I haven't bored you too much at this point, is the GoSlims. This is going to take a little bit to go through, so bear with me. And if any of this confuses you, please feel free to ask questions at the end. So back at the home page, go ahead and click Explore Biology, and then it will take you to the GoSlim setup. But before we do that, let's go over what a GoSlim is and why they are useful. So GoSlims are cut down versions of the Go ontologies containing a subset of the terms in the whole Go. They give a broad overview of the ontology content without the detail of the specific fine-grained terms. So we're going for Go terms that are not too specific, but specific enough to get us a general overview. And they're very useful for giving you a summary of the Go annotation to a particular genome, a microarray or a cDNA collection, when you need these broad classifications to help you figure out what a gene product will be doing. And GoSlims are always created by users according to their needs, and they can be specific to a particular species or to a particular area of the ontology. So if you only want to focus on zebrafish, you can use a GoSlim specifically designed for zebrafish or for yeast. If you're interested in Candida albicans, not Candida albicans, but yes, Candida albicans. And let's say you're only interested in biological process, so general biological process terms. So you want to get an overview of the proteome for transcription and for metabolism and a few other areas of interest. So this is the actual GoSlim setup page. So we offer an option to go for predefined GoSlims, input your own, or select from the basket. That's why I was highlighting the basket option before, because if you see a Go term you think would be good for your Slim, you can throw it in the basket, then pull it up over here. Or if you have a list of Go terms in a separate file, you can copy and paste them and use the input your own option. Or you can use the predefined ones. And if you click on that drop-down menu, it gives you a few options for some of the ones at the Go consortium, the ones responsible for maintaining these GoSlims, have defined. And for in this case, we're going to be using the fly-based GoSlim. Once you put in the terms, you should get an option for gene product IDs. So if you want to Slim for particular gene products, you can do that. So let's say you did a mass spec experiment and you identified a bunch of proteins from a cell that you've treated with a drug, and you want to see what areas of what functions of the cell have been impacted. So you can take your entire list of identified proteins, find them in Uniprots, or if they're in particular, if they're proteins, if we're doing mass spec, throw them in here. And you can further add a filter for restricted by taxonomy. So if you know you're only working in, let's say, Drosophila melanogaster, this is where you can filter that as well. And you know your terms have been added when you see this over here on the bottom where it says your selection, 45 terms, and it breaks it down. And then once that's all set, you hit apply. So if you're making your own GoSlim, the terms need to be independent of each other, meaning that if you were to look at the ancestry tree, and I can't emphasize this enough when you're setting up a GoSlim, is the terms that you've selected, if you were to look at them, in this case, we're looking at the fly-based one again, we are interested in general processes, nothing too specific, but they have to be independent of each other. So we can already see here that, for example, all the ones highlighted in yellow are the biological process terms that we're interested in, but none of them are part of are not directly related to one another. Because the way a Slim works is that if, let's say, we look at the DNA metabolic process, any child of DNA metabolic process, when you Slim, any annotation to a child term, a DNA metabolic process, when you Slim, will map up to DNA metabolic process. Same thing with protein metabolic process and gene expression. So you want to keep the selection of terms rather generic in a way that you don't accidentally pull up annotations within the same branch, as well as you're able to pull out all the terms that would make that particular Go term meaningful or that particular biological process meaningful. Because, for example, cell cycle includes a lot of different processes. So you want to go for a term that's going to encompass them all to make sure you get as many annotations as possible to make your SLIM meaningful. So if you guys went ahead and did this along with me, if not, don't worry about it. I did the fly-based GoSlim annotation, and I restricted the filter to flies, so to Drosophila melanogaster. What you see here is an example of the output. So you see the Go term that we used in the SLIM over here, and the original Go term refers to the Go term in the annotation. So what you end up seeing then is that for cell cycle, for example, we pull down an annotation to mitotic cell cycle for this, this gene product over here, VTD. Then we also see that it's involved in this mitotic cell cycle. It was determined by a mutant phenotype from this particular paper, and this was done by Flybase. And if you're interested to know and have an executive summary of what your SLIM has pulled out, you can click on the statistics tab, and that'll give you this, and it'll tell you that you've pulled out this many annotations to this many distinct gene products, which is pretty good. You should have a rough idea when you're doing a SLIM for an entire proteome, how many proteins are there. So the distinct gene products will give you a rough idea of whether you've pulled them all out or not. You can now also export this to get your SLIM summary, your Go ID, sorry, not your SLIM summary, but your Go IDs that you've pulled out, the aspects, the percentage of the coverage in each aspect from your SLIM, the evidence that was the percentage of evidences, the percentage breakdown of evidences, references where they came from, and then also who made the annotations if that's important to you. And just some tips that PalmBase has been kind enough to lend to us to use for this presentation. So when you're using a GoSlim, first one, when creating a SLIM for the entire genome, make sure that it covers as many annotated genes in your set as possible. I know I've covered some of these already, so bear with me. You should be aware of how many genes are annotated but not in your SLIM and how many are unknown. How many genes are annotated but not in your SLIM and how many are unknown in terms of how many genes have no annotations at all, but you know they are part of the proteome of your organism. For display purposes, you want to keep the number of terms as small as possible to convey your results. So if you're using this for a paper or a presentation to your department or your group, you don't want to have 45 terms in biological process, another 90 in molecular function, and another 60 in cellular component. It's going to make it way too confusing. You want to keep it concise. So that's why we use the Flybase one. It's nice and simple. It's only 45 Go terms in the initial SLIM. And if you're using your SLIM for data analysis to summarize, let's say Go term enrichment, which again is a completely separate topic, but if you guys are interested in a webinar on that, let us know. You should ensure that the terms are specific enough to demonstrate their relevance to the biological topic of interest. So in this case, PalmBase gave the example of lumping all genes involved in transport may mask overrepresentation of transmembrane transport versus underrepresentation of vesicle-mediated transport in your results. So you want to make sure that when you make your SLIM, the categories you've chosen represent your results effectively. And finally, the most current implementation of software to create GoSlims includes the regulates relationship by default. So not all the time, not every older GoSlim softwares will not include the regulate relationship. They'll just include the occurs in, part of, and enables. But by having the regulates, it actually allows you to include more annotations that might be able to pull out gene products that may be regulating the process that your drug may have affected. Finally, the basket. How to make this basket useful for you and when you're using QuickGo. So you can add Go terms to your basket to generate Go term comparisons, as well as to generate custom GoSlims. And I've already talked about the custom GoSlims, so I'm not going to go too much into that. So when you click on the basket, it shows that your basket is empty. You can do keyword searches in the search bar, or look at the annotation table and select Go terms there and add them to your basket, and it'll all appear here. And when you add them in, so in this case, I added 4 random ones in here. It gives you several options. You can say you can filter the annotation set with those terms. You can export those terms to a new file. Or you can continue adding. Or if you click on this little button here, that's the ancestry tree. It allows you to actually compare those Go terms to see how they relate to one another. And we see in particular that these Go terms are related to one another, but through different relationships in the ancestry tree. So this allows you, if you're trying to come up with terms for a SLIM, to quickly take a look at what you've picked out so far and how well it's going to work in your SLIM, or if it's not. So if you see, for example, that you have signal transduction and cellular response and transforming growth factor beta, this is not going to quite work out. Well, it'll work out decently well. They're not directly connected, but you may end up having some annotations that overlap between the two. But for example, gastrulation and signal transduction are not related to one another, so these would be excellent for a GoSlim. But if you were to have this transforming growth factor beta receptor activity down here, this would be a problem for your GoSlim, because your annotations would slim up to this term as well as this term and signal transduction. So it would create a lot of problems for you, and you wouldn't get meaningful results. And of course, if you need any additional help, you can check out our FAQ page on the help section, or contact us directly through the contact page with your query. We do answer our emails within a day or so, or usually within the same day, depending on how busy we are. And of course, if you have any questions or comments, you can contact us at QuickGo. So that's the links for the first link. You can go to our group page at ebi.ac.uk/Goa. You can email us directly at goa@ebi.ac.uk, and you can also follow us on Twitter at QuickGo_EBI.